This video tutorial is about enzymes. Okay, so what we're going to do is we're going to use an envelope and we're going to put together some notes of key terms and then we can keep them for revision later on. Okay, so let's start with what an enzyme is. So, an enzyme is a biological catalyst. It speeds up a reaction, it remains unchanged and can be reused afterwards. That means the body doesn't constantly have to make enzymes all the time because they can be reused for the same reaction many times. The next thing we have to do is we have to recap about protein structure because all enzymes are proteins. So all proteins have a primary structure, which is where amino acids join together using peptide bonds. They then go into secondary structure, which is where those chains of amino acids are folded into alpha helix or beta pleated sheet and are stabilised using hydrogen bonds. Tertiary structure then is where an enzyme will always be. It's what it relies on because tertiary structure gives enzyme a 3D shape. Okay, so most enzymes are globular proteins, so they become ball shaped. And the bonds which help to stabilise this level are still peptide bonds and hydrogen bonds, but also ionic bonds, hydrophobic interactions and disulfide bridges. Okay, so some key terms with enzymes. Firstly, all enzymes have an area on their surface where their substrate fits, okay, and that's called the active site. So if we imagine that this is our globular enzyme, then that area there is the active site. And that will be complementary in shape and charge to the enzyme substrate, which is what it works upon. So for example, if this is the substrate and this is the active site, complementary means that they can fit together like that, okay? All enzymes are specific or substrate specific and they tend to work just on one specific substrate. So for example, amylase, okay, is an enzyme which works on amylose, which is a part of starch. And quite often you can tell what the substrate is that an enzyme works on because the substrate is the first part of the name of the enzyme, and all enzymes tend to end in ASE. So for example, lipases are enzymes which break down lipids. Okay, so there are two theories about how enzymes works. The first one you might have come across at GCSE and it's called the lock and key model. Okay, so as we said, all enzymes have an active site where their substrate fits, and what happens is there has to be frequent collisions between the enzyme and the substrate. They have to come together and those collisions have to be successful, which is when the substrate collides with the active site of the enzyme like that. That forms an ESC or an enzyme substrate complex. What happens then is enzymes lower the activation energy, that's the energy needed for a reaction to take place, and therefore when the substrate comes away it will have formed into product, and that product will no longer be complementary in shape and charge to the active site. Okay, so that's the lock and key model. The next model that we need to have a think about is called the induced fit model. Okay, the induced fit model is very similar to the lock and key. It involves a change in shape. So what we're saying is, actually, when the enzyme and substrate come together, okay, the enzyme squeezes around the substrate to make an even better fit. By doing that, it stresses the bonds of the substrate, making it even easier to break down the substrate and to form products. So by that, it's lowering the activation energy. So that is the induced fit theory. The next thing that you need to do for your syllabus is to look at how different factors influence enzyme catalyzed reactions. So look at the effect on the rate of reaction. So the first thing we could look at is enzyme concentration. So as we see here looking at this graph, as we increase the enzyme concentration, we increase the rate of reaction. That's because there are more active sites, which means that we can get more enzyme substrate complexes after those successful collisions and we'll get more product formed, and that's how we track the rate of reaction. Eventually though, that graph will level out and we can carry on increasing enzyme concentration, but the limiting factor there would be substrate concentration. You have to have the substrates to form the product. So the next thing we can look at then is the effect of substrate concentration. So we get a graph of two halves here, part A and B. As we increase substrate concentration here at A, we see that the rate increases, and that's because there are more frequent collisions, more enzyme substrate complexes and more product formed, the more substrate we have. Eventually though, the graph will plateau out at B. That's the level that we call the V max. What's happening there is the enzyme is working at its maximum velocity. That's where we get V max from. And what's the limiting factor is the number of active sites. Eventually we'll get to a point where there aren't enough active sites which are free for substrates, and therefore we can't get any more product formed. An analogy that you could use with that is if you're going to do your shopping. Okay, so as the shopper, you are the substrate and the person at the checkout is the enzyme. Now, if you have 10 people on the checkout and 10 people doing their shopping, then one person can move through every checkout and 10 people can move through all at once. If we then have 20 substrates or 20 shoppers, but still only 10 enzymes or 10 people on the checkout, you can still only get 10 people moving through at one time, and that's because it's the enzyme, or in our case the checkout, which is the factor which is limiting. You have to have a free checkout or enzyme's active site in order for a reaction to take place. So the next two factors are environmental factors that you need to have a look at. Firstly, the effect of temperature. With temperature we tend to see this quite non-symmetrical graph. So at point A here we can see the increase in the temperature, we get increased kinetic energy, so our enzyme and our substrate are moving around. That means that we're going to get an increased frequency of collisions, more enzyme substrate complexes, and therefore we get an increased rate of reaction. That happens up to B, which is the maximum rate, and that happens at the optimum temperature. Now, every enzyme has its own optimum temperature, but most of the ones in the human body are around 40 degrees C, because that's just a little bit higher than our internal body temperature at 37. We can see though that beyond the optimum, here at C, we get a very, very fast decrease down to no rate at all. So the reaction stops as soon as we go over the optimum. That's because with the increased kinetic energy, what happens is it breaks the hydrogen bonds, okay, which remember help us stabilise our enzyme in tertiary and also secondary structure. So we lose that 3D shape. We don't have the active site anymore, so the substrate cannot join to it, and that means our enzyme has denatured. So at high temperatures, enzymes denature, and therefore they're no longer functional anymore. Okay. The next environmental factor that we need to look at is pH. Now we can see here a much more symmetrical graph around part A, which is the peak of the graph, where that's the optimum pH. So at the optimum pH we get the fastest rate of reaction. As we move, slight changes, either increasing or decreasing pH, we see the same thing, which is a decrease in the rate of reaction. What's happening there is we're getting a change in the number of hydrogen ions in the environment, and that changes the charge on the enzyme's active site. So it could be that we don't get quite the same complementary fit, so the rate of reaction decreases. As we go down to point C, which is extremes of pH, either very alkaline or very acidic, what happens there is that though the effect on the charge is that it breaks those bonds which are associated with charge, which are ionic bonds. Remember, our ionic bonds were at our tertiary structure level, so again we lose 3D shape, and therefore we lose the shape of the active site, and the enzyme is denatured by extremes in pH. The last thing that we need to look at is the effect of inhibitors. There are two types of inhibitor. There's competitive inhibitors. Okay, competitive inhibitors bind to the active site. So if we have a look at our Play-Doh enzyme with its active site here, okay, remember this is our substrate, complementary in shape and charge. So an inhibitor is any molecule which is similar in shape to the substrate and therefore can fit into the active site and bind. So if this is our inhibitor molecule, there it's bound to our enzyme. By doing that, our substrate molecule can no longer bind. So that's going to decrease the rate of product formation and decrease the rate of reaction. The only thing that we can do is increase the number of substrates, and therefore if we had more substrate, then we could outcompete the inhibitor, and therefore it would be the substrate which would bind with the active site and still form product. Okay, so those two are in competition. The last type of inhibitor is a non-competitive inhibitor. This time it doesn't bind to the active site like the substrate does. It binds somewhere else. So if we imagine that this is our non-competitive inhibitor and it binds to the allosteric site at the back of the enzyme in this case, by fully binding, so this time if we press our Play-Doh enzyme onto the brick, what we see happening is we see that the non-competitor inhibitor changes the full shape of the enzyme, including the active site. So even if the non-competitor inhibitor unbinds, our enzyme has still changed shape, the active site has changed shape, and it can no longer fit with the substrate. So here, that's going to decrease the rate of reaction probably down to 0. Okay, so we've made some notes about enzymes, so I'm going to keep those and put them away for revision. Okay, that's there. And you're going to have a question and an answer for you to have a look at on enzymes on the internet.